not least, the innermost layer, we've got that neural layer. This is the retina that we keep talking about. Okay. Now, we got two parts to the retina. You've got your pigmented layer and you've got the neural layer. So first we've got the pigmented layer. This is the thinner, more outer of the two layers, which is kind of confusing because this is the innermost layer and we've got a double layer, and so we've got an outer and inner, so that gets a little confusing. So the pigmented layer, we're going to help absorb light that passes through the neural layer and prevent it from bouncing back. Okay, so basically we want to, so if you think about like a reflection in a mirror, we don't want the reflection itself, we want it to be absorbed. Okay, and so that's what the pigmented layer helps with. This is also where we store vitamin A. We have beta carotene as kind of a precursor to the vitamin A that helps with normal function of your photoreceptors. Now we're going to talk a little bit more in detail about photoreceptors here in a little bit, like literally right down here. Okay, so the neural layer, all right, the thicker layer, the innermost layer, this is where you do find those photoreceptors that we just mentioned. You have two kinds of these. We have rods. Okay, the rods are found on the lateral portions of the retina, and as you move towards the center of your eye, your rods decrease in number. So rods, lateral portions of your retina, so on the sides. Okay, so that's number one. Number two, you have cones. Okay, we have a high concentration of these cones in a particular spot known as the macula luteum, which basically means yellow spot. It's kind of funny. This is where images are formed in your eye. Okay, the highest concentration of cones is at the very center of that. We call that the fovea centralis. Okay, so within the yellow spot, we have the fovea centralis. Okay. This is where you have the clearest, sharpest vision of anywhere else in your whole eyeball. Okay. Now, where's my thing? Here we go. Oh, here we go. Okay. So, like, why do we have two different types of photoreceptors? Because they do two different kinds of things, obviously. Okay. So, even though they do two different things, they do kind of work in a similar fashion. So, both your rods and your cones, okay, are going to send sensory information along what are known as bipolar cells. Okay, this is just a specific type of neuron. Bipolar indicates that we have two poles, okay, which would totally be true. The bipolar neurons will then send information along your ganglion cells. Okay, your ganglion cells are going to fuse, and that's what's forming your optic nerve that you are already familiar with. Now, the optic nerve itself, it basically comes out the backside of your eyeball, so the posterior side of your eyeball. It has no photoreceptors itself, okay? And so where your optic nerve comes out the back of your eyeball, we call this your blind spot or your optic disc, okay? So its only function is to transmit the signal received from the bipolar cells through the ganglion, right? So we're taking the information through the optic nerve to your brain, all right? So we're not receiving information here. Now, we mentioned that we do have both the rods and the cones. So we've got two, and I told you they do different things. So here's what's going on. First of all, you have more rods than you do cones, okay? These are used for your peripheral vision. Now that should make sense because the previous slide we said that the rods basically sit on the lateral aspect of your eyeballs themselves. Well, what is your peripheral vision? It's what you can see out of the sides of your eye. This helps us create a fuzzy, kind of grainy black and white image in low light, okay? And it helps us detect movement in our peripheral vision, okay? Now, if you don't have a lot of rods, if your rods get damaged, if you have a lack of vitamin A, any of those problems, you could suffer from night blindness, okay, which is fairly self-explanatory. You have difficulties seeing at night because there's not a whole lot of light at night and so this peripheral vision kind of goes away, all that kind of stuff. Now, the cones. Cones are for color. Okay, cones are for color. This helps us produce a sharper, colorful, clear image. We see in we have three types of cones, basically. We've got blue ones, green ones, and red ones. Okay. We have overlapping abilities to see in between the reds and the blues and the greens. So all the other colors of the rainbow, these kind of work together for us to be able to process the in-between shades. Okay. Now, some people do suffer from color blindness, which basically means that you are unable to see one or more colors. Okay. This is a genetic disorder. Red-green color blindness is the most common. It's not the only type of color blindness, it's just the most common. And this, because of how it is inherited, it is much more common in males. Now, again, that doesn't mean females can't be colorblind. It's just less common for females to be colorblind. All right, here we go. So just to kind of make sure we are on the same path, you can see, where's my thing? Here we go. All right, so your optic nerve in the back, coming out the back of your eye, we've got the fovea centralis, that tiny little dot. Okay, so that's where we've got all those cones at. So here's our pathway. So let's wait for it to start over. Here we go. Light's coming in. These are the rods and the cones. Okay, these are your rods and your cones. These are the bipolar neurons that we talked about. So we said one pole, two pole, bipole. All right, we're going to transmit the signal through the ganglion cells, and then this would fuse and become your optic nerve, and we would send the information to the brain. Okay, and again, here we can see these bipolar neurons. We've got the bottom part of it here, but you can see the other side of it there. Okay, all right, how's everybody doing? All right, we talked about cones being used for color detection. We've got the blue ones, the green ones, the red ones. I told you there's a lot of overlap, so we can see the in-between colors as well. And again, if you suffer from color blindness, so red-green color blindness, if you don't have either one of these cones, you would be unable to detect, these particular shades. We still do the purples and the blues and stuff, but these other colors would be difficult for you to detect. There are colorblind tests that you can take. These are called Ishihara tests, and each one of these does show a different thing. So this is actually the number 58. This is 18. This one, this is actually a letter E. This one is a... Some people say it's a 17. Some people say it's a 12. This little foot is a little short, so I'm not going to argue if it's a 17 or a 12. This is a 25, this one's 45, this is 6, this is an 8, 56, and 29, okay? So, just the difference of what you can see, what you can't see. So if you saw, if you thought this was just a 5, or if you thought this was just an 8, that tells your doctor something. So there you go. All right, so we've got a couple of eyeball structures left to chat about. We've got your cavities and your chambers. So your eyeball, we said it was spherical. We said it was hollow. I told you it wasn't completely empty though. You do have these cavities and these chambers. You have both an anterior and posterior cavity. Okay, and your anterior cavity is then broken up into your anterior and posterior chambers. Okay, so it gets a little confusing. We'll go through all the words and then we'll show you some pictures, okay? So your anterior cavity, this sits in front of the lens, okay, and your ciliary body. Specifically, the anterior chamber is between the iris and the cornea, and then the posterior chamber would be between the lens and the iris, okay? And both of these chambers are filled with fluids, okay? We call this your aqueous humor. Okay, pretty similar to your blood plasma. This helps you maintain your intraocular pressure. Okay, pushes against the cornea. This helps make sure that light is being bent in an appropriate fashion as it goes through the lens to help with focus. It also helps make sure your eyeball is nice and round. Okay. Now, your posterior cavity, the larger one, this sits completely behind the lens. Okay, so if you think of the hollow space in your eyeball, you're probably thinking of the posterior cavity. Okay, this is actually filled with a different kind of fluid. This is vitreous humor. This isn't quite as fluid-like because we've got a lot of collagen within it. This is a little bit more jelly-like, just a little bit more jelly-like. This presses against the retina and the choroid layers. This also helps maintain the majority of your eyeball shape since this is the larger cavity. Okay, it plays a much bigger role there. This is also helping transmit the light to the retina. Okay, so again, I know the words are a little confusing, so let's look at a picture. So your anterior cavity, okay, remember your anterior cavity, our dividing line is technically the lens. Okay, so between the lens and the iris, we've got that posterior chamber, and then the anterior chamber would be from basically the iris to the cornea. All right, so we got a little fluid in here, and we've got a little fluid in there, that aqueous humor. Now, this is the posterior cavity. Again, usually when people think of the space in their eye, they're thinking of this big space back here. This vitreous humor would be living back here. Okay. All right, then we have the lens itself. We've got this located posterior to the cornea. We're going to hold it in place by the suspensory ligaments, which are coming from the muscles in the ciliary body. We've already talked about the ciliary body. Your lens is convex in shape, which means it pokes out on both sides. Okay. This helps with refraction and focusing of the light as it comes in through the lens, and we're going to send that light back to the retina. Unfortunately, as we get older, the lens becomes less flexible. Okay, this tends to be one of the reasons that people start to need some sort of reading glasses or bifocals when they start to get into their 40s. Okay, that's just a general rule of thumb. Okay, your lens should be transparent because we want the light to pass through it. If your lens starts to become opaque or cloudy, then you are starting to suffer from cataracts. Your lens can be replaced, so we can cut it out and substitute an artificial one in there that is transparent. If light is not refracted properly as it moves through the lens and the cornea, then you probably suffer from something called astigmatism. Okay, and that doesn't necessarily have to be in both eyes. So, for example, I have an astigmatism in my right eye, but not my left eye. It just is what it is. Okay, but that's what's going on, is that you're not refracting your light properly. We mentioned cataracts. We said that the lens is going to become more opaque, and so this would be your normal eye. This would be an eye suffering from cataracts. Okay. This is the most frequent cause of blindness, so if you do not have surgery to correct this, once it gets bad enough, light won't be able to move through your eye at all, and you will become blind. Now, glaucoma is also a pretty common eye condition. If the aqueous humor in the anterior cavity of your eye, if it does not drain properly, then the fluid will build up. It can actually build up into both the anterior and posterior chambers of the anterior cavity. The more fluid you have, the higher your intraocular pressure. Okay, if you put too much pressure on your eye, then you can damage your retina and the optic nerve themselves, and so you can see what a normal eye is supposed to look like, and you can see what a damaged nerve kind of looks like, and you can kind of also see what somebody with glaucoma would see. So normally you should just be able to see apples and a little bit of shadow from the apples themselves, but if you suffer from glaucoma, then your surroundings become blurry, things like that.